Hello, my name is Sarah Munro. I'm with the National Institute of Standards and Technology, and I'm here today to give a lecture on work that I've been doing at NIST to develop RNA controls and analysis methods for trustworthy genome-scale measurements. So today we'll cover 4 topics. We'll start with a little bit of background on gene expression measurements, and then I'll talk about work we've done hosting the External RNA Controls Consortium, the ERCC, to develop RNA spiking controls. And then we'll cover the ERCC dashboard analysis tool to use for method validation in gene expression experiments, and then recent work that we've started to develop an updated suite of RNA controls in a project we call ERCC 2.0. So to begin with gene expression experiments, let's talk about what gene expression is. An example to look at here would be a caterpillar and a butterfly. They have identical genomes. The differences between these organisms arise from when the genes are expressed and which genes are expressed. And this type of application of measuring gene expression from an organism with the same genome and how those genes are being expressed at different times is a critical application of quantitative biology in the post-genomic world. And the Human Genome Project was hugely enabling because it allowed people to develop the microarray technologies to make these types of measurements. And so when people do gene expression experiments, they started off with microarray technology, which came out of Ron Davis's lab here at Stanford University. And in these microarray measurements, cDNA molecules representing the RNA in your samples, say you're comparing a butterfly and a caterpillar, those cDNA molecules are hybridized to probes, and then gene expression measures are derived from fluorescence signal intensity. Now, newer technologies are coming out, such as RNA sequencing, and in these sequencing measurements, the cDNA molecules are actually sequenced, and then the gene expression measures are derived from the number of times a sequence is counted. So when we do these gene expression experiments, how do we know that we have trustworthy results? We're simultaneously measuring tens of thousands of RNA molecules in our samples, and how do we know if we're getting it right? Well, back in 2003, when DNA microarrays first came out, Maggie Kam, a scientist at NIH, published this seminal study where they compared different microarray platforms from different technology providers and found that they had wildly different results. This Venn diagram really caused a lot of dismay in the community, in particular for technology developers. And they decided that they wanted to work to find a way to make these measurements reproducible, because clearly when you have only four genes that showed agreement across these platforms, you're having irreproducible results. So that's when the External RNA Controls Consortium came into being. This consortium was initiated by industry. The first person to call for it was actually Janet Warrington, who was at the time VP of Clinical Genomics at Affymetrix. And then the consortium was hosted by NIST because NIST has the capacity to convene all interested parties to develop standards in a pre-competitive space. There were more than 90 participants, including all technology developers of microarrays and other gene expression assays. And the goal of the ERCC was to develop external RNA controls, or commonly called spike-ins, to put into gene expression experiments at the beginning of the measurement process so that you would have these controls that went through with your sample RNA molecules all the way through the measurement to the final results. And the product of the ERCC is this NIST Standard Reference Material. We call it SRM 2374. This is a DNA sequence library, and each tube contains a unique ERCC control sequence in a DNA plasmid backbone. The controls were intended to mimic mammalian mRNA. And to produce the RNA controls, you do in vitro transcription from the plasmids and then combine the controls into mixtures. And this is an example of how you would do that. We choose different plasmids from the DNA plasmid library, and produce RNA transcripts from each of those plasmid types, and then pool them into mixtures with known abundance ratios. So for example, you can see here that in the tube number 1 here, we have 1 blue transcript, and tube 2, we have 3 blue transcripts. So that's a 1-to-3 ratio between these two mixtures. And you can do opposite ratios, such as 2-to-1 for the green transcript. And so what that allows you to do is develop built-in truth with known ratios. And then we can use these controls in our RNA gene measurements in gene expression experiments. So, for example, say we have 2 different mice, one that's been treated and one that has that's the control, multiple replicates, of course, of these mice. And you can see that there's differences in these mice phenotypically, but when we look at their RNA, it clearly is just a pile of RNA molecules. But if we add in the RNA controls, we can see that we have known abundance ratios between the treatment and control samples now. The RNA controls then go through the measurement process along with the sample RNA molecules, and we obtain expression measures at the end of the process and perform statistical analysis. And this process takes multiple steps. Many people and laboratories are involved, and it can take days to weeks. And so it's really important to know if you've done this right. So the example type of gene expression data that you would get from this is shown here, where you have a table essentially of data with all of the different transcripts that you're looking at from your sample, and then the ERCC control transcripts are also included in your results, your data. And so for example, this data might be from an RNA sequencing experiment where these are actually counts. But what's important to note is that in these experiments, we're really asking about the ratios across the 2 sample types, because when you're measuring tens of thousands of molecules at once across a broad dynamic range, you can't necessarily measure an actual abundance. We don't really know with certainty about how many molecules are in each of those sample types. So what we do is look at ratios, and this is important. The ratios are stable when we compare them across the treatment and control samples. And so here I'm showing the ratio data, and then we do statistical testing to obtain p-values to say whether or not this is actually a true difference that we're seeing in a sample or not. And then with the ERCC controls, we have true positive and true negative ratios. And so we have this built-in truth that we can use to understand the performance of our experiment. So now I'll talk about the ERCC dashboard analysis tool that we use to actually look at this data and do statistical analysis to understand how well we did with our experiment. And so the ERCC dashboard is a software package written in R. It's available from Bioconductor. It's been in Bioconductor for the past 1.5 years. And it's also available from the NIST GitHub site. And the idea here is that this tool is open source and open access. It can be used in other analysis tools and pipelines, and it can be adopted and has been adopted into commercial software, and is used for internal testing at technology development companies. So the idea here is that you can use these on any type of data, again, microarray, RNA sequencing, or new data that comes along in the future. And so the ERCC dashboard produces these 4 ERCC dashboard figures that tell us about the performance of our experiment. And this package was developed as part of the FDA-led SEQC study, along with ABRF partners. ABRF is the Association of Biomolecular Resource Facilities. The technology-independent ratio measures that come from this tool can be used to assess differences in performance across experiments, across laboratories, and between different types of measurement processes. And this work was published back in 2014 in Nature Communications. So in this study, to develop the ERCC dashboard, we used the Ambion ERCC ratio mixture product. This is a commercially available product made by Thermo Fisher that is, they're essentially mixtures of 92 of the ERCC controls. Imagine these are 2 of the tubes that I showed earlier. So these are prepared using the NIST Standard Reference Material Library to produce these controls. And so this particular design has three differential ratio subpools, where we have controls that are at a 4:1 ratio, a 1:2 ratio, a 1:1.5 ratio, and a 1:1 ratio. The 1:1 ratio is very important to have because that is our true negative, where we know that there should be no difference across the samples. So we can also look at how this data looks across the dynamic range. So this is what the design was of the ERCC controls for the Ambion mixes, where we have a 2 to the 20 dynamic range. And again, we have these different ratios that are shown here in log 2 space. So for these SEQC RNA sequencing experiments, there were 2 types of experiments that we looked at, and I'm showing them here on the left and on the right, the rat experiment and the interlaboratory experiment. So with the rat experiment, the spikes were added to biological replicates. So each treatment and control rat received spike into the total RNA samples. And then those sample replicates went on for RNA sequencing. In the interlaboratory experiment, the design used human reference RNA samples that are commercially available, and a large batch of each type of RNA was spiked. And then those samples were distributed into many, many aliquots and shared amongst multiple laboratories. So now I'll go through and discuss each of those 4 figures for the ERCC dashboard for these 2 different types of experiments. So one might ask, when you're doing an experiment like this, what is the dynamic range of my experiment? And so you can see here, for the rat experiment, there's a slightly truncated dynamic range compared with the interlaboratory experiment. And the reason for that is that in the rat experiment, typical sequencing depth was done where there were 40 million sequence reads per replicate. And in the deep sequencing experiment that was done in the interlaboratory study, there were over 260 million sequence reads per replicate. So it's quite a bit. And so in the interlaboratory study, they were able to capture the full 2 to the 20 dynamic range of the design. Now, the next thing one might look at is the ratios in these experiments. In the previous slide, we were looking at signal versus abundance of the molecules. But now we're going to focus on the ratios of the molecules in the samples. And so here we're looking at ROC curves. And in these, we're looking at each of the true positive types of RNA controls compared to the true negative pool, the 1:1 controls. And you can see that there are these ROC curves are pretty great for the rat experiment, and also they're pretty good for the interlaboratory experiment. One might be tempted to compare these 2 and think that there was a better result in the interlaboratory experiment, but I would caution folks to look at the gray box down here, where the area under the curve is shown for each of these experiments. And we also show the number of controls that were detected versus the number that were spiked into the experiment. And in the rat experiment, less controls were detected than were spiked, 16 out of 23 for each of the differential ratio pools. And then for the interlaboratory experiment, 22 or 23 for each pool were detected. So AUC, you can see, has some limitations. It's a single summary statistic, but it is reasonable, and it's something that folks are using consistently in the field. But we wanted to try to evaluate our diagnostic performance as a function of abundance, and so we developed this new metric that I'll describe now. So first, let's look at what the ratio versus abundance data looks like. This is the rat experiment, and this is looking at the ratio versus abundance plot. So if you recall the design plot from earlier, we have the 1:1 controls were here at 0 on the log 2 scale, and then we have the 4:1 controls were up here at 2. And so this is actually the real data now that we're looking at, and then the endogenous transcripts in the samples are actually shown here in gray. And so what we really want to understand is how good are we at seeing differences from no difference, right? So when actually can we detect a ratio difference reliably, right? You can see here that the green points and the red points and blue points are all getting muddled down at low abundance levels, and so it's really important for folks to know essentially how low can you go in their experiments. And so we've developed this new metric called the limit of detection of ratios. And so here you can see the LODR plot for the rat experiment, where we're modeling p-values as a function of average signal in the experiment. So this is the average count data, and so we're increasing in counts as we move from left to right across this axis. And then the p-values from differential expression testing are gradually becoming smaller and smaller as we move down the vertical axis. And so, as one would expect, for the one-to-one controls, the true negatives, where you had the green ones, where there shouldn't be any difference, the p-values are all very close to 1, which is the correct result that you should see. And then for the other types of controls, the 1-to-2 controls and the 1-to-1.5 and the 4-to-1 controls, you can see that the p-values are getting smaller and smaller. And so what we do to determine LODR is we find a p-value threshold based on our chosen false discovery rate. And so here in this experiment, the false discovery rate is 0.1. In the ERCC dashboard software package, the default false discovery rate is set to 0.05, which is commonly used in the community. And so what we do is we estimate LODR for each of the differential ratios from the intersection of the model confidence interval upper bound. So here, this top part of the confidence interval around the 4-to-1 model function, we find where that intersects this black dashed line that represents the p-value threshold. And that count value is our LODR. And the dashboard automatically calculates this value for you and annotates it in a graph, or in a table at the bottom of the graph. And so for this experiment, for the 4-to-1 ratio, we have an LODR estimate of 26 average counts, and we also determine the 90% confidence intervals on this result and report those as well. And so this is really useful because we can use LODR to specify what our confidence is in finding differentially expressed transcripts. And so we can say we have a 90% chance of having a false discovery rate that's less than 10% when we're above 26 average counts in this experiment. And that's useful to be able to give quantitative results. And so this can be used for scientists to design new experiments. They can try to increase signal for their endogenous transcripts in the sample to be above an LODR estimate that they're targeting. So I'll show an example of that here, where I've annotated this figure with that 4-to-1 LODR that we estimated. And then we can see that this region up here is where we're above the LODR. And let's say we're sort of interested in transcripts that are shown here with red asterisks. If we were to increase sequencing depth, then those endogenous transcript ratio measurements might be shifted above LODR. And so, again, just to show you what this LODR estimate looks like for the interlaboratory experiment in comparison to the rat experiment, you can see that there's actually, the p-values are much, much smaller in this experiment because the sample types are very, very different. So it's much easier to find differences in this experiment than in the rat experiment, where most transcripts are not differentially expressed. And that's also demonstrated in this final figure from the ERCC dashboard, where you can see this gray cloud of transcripts in the rat experiment is showing a much more typical shape that one would see in a biological experiment, where there's not a huge amount of scatter. We don't have very extreme ratios. In the human reference RNA samples that were used in the interlaboratory experiment, the spread of the ratio data is much, much greater because there are extreme differences between these samples. So this figure I've also annotated where that 4:1 LODR is, and we can see how the endogenous samples actually relate to LODR, which is also annotated on the figure where the solid points for the ERCCs are above the LODR and the open circles are showing ERCCs that are below the LODR estimates. So another piece of that particular figure, which was shown with the error bars, is looking at how much technical variability and bias is there in our ratio measurements. And so one can see here in these distributions of the ratio measurements for the rat experiment and the interlaboratory experiment, there's actually significant ratio bias for the interlaboratory experiment, where the design value is here, shown at log 2, and then the actual result is shifted significantly. And there's a reason for this in this particular experiment, and that is mRNA fraction differences, and I'll describe that briefly here. This is actually really important, and it's actually really cool. It's a feature of the ERCC controls that you can actually see this mRNA fraction difference between samples. And so if you imagine you have sample 1 and sample 2 consisting of message RNA, mRNA, and ribosomal RNA, and of course many other RNA molecules, but this is a very simplified cartoon here. And we add our spike-ins, and we add in the same amount of spike-ins to our samples. And then once we do the mRNA enrichment process, we remove all the RNA except for the mRNA that we're interested in this case. And then that actually shifts the relative amounts of spike-ins in each of those samples. And so that skews our ratios. So what we thought was a 1-to-1 ratio is now shifted, and the same thing with all the other ratios. And so that's important to note that this type of shift in the ratios across the samples is probably not just happening to the ERCC controls, it's probably also happening to endogenous samples. And so it's good to know what this shift might be so that we can potentially correct for it. And so to summarize what you get from the ERCC dashboard, we have these 4 main figures with dynamic range, looking at diagnostic performance with our ROC curve and the AUC statistic. And then we also have this new metric that's been developed called the limit of detection of ratios. And then also we look at the ratio abundance plots to see variability and our bias, which is annotated here in this gray inset box. And we can compare how our LODR estimates from the ERCCs how they relate to the measurements for the sample transcripts. So that's great. You can use the ERCC dashboard for an individual experiment, and you can also use it to compare experiments across laboratories. And this is where you have the ability to evaluate reproducibility across laboratories. And so this is from the same paper. When we looked at the interlaboratory data, you could see examples like this, where the results shown on the left are a laboratory that had good performance, and then on the right here we have poor performance, where it's really difficult to discriminate true positives from true negatives in this experiment. And so there were actually 12 different laboratories that we compared in this interlaboratory analysis, and I've color-coded them here to show you the differences. Each one had a distinct measurement process. And so the sequencing platform used were either Illumina, LifeTech, or Illumina in these different laboratories. And then the laboratories each used a different type of poly-A selection protocol. There were 2 different poly-A selection protocols, one from Illumina and one from Life Technologies, and then a ribosomal RNA depletion protocol. And so an example of what we found when we compared LODR across all the laboratories was that it was consistent across 11 out of the 12 laboratories. And that was reassuring to see that we are able to get consistent results with these samples. And we found that Lab 7 was an outlier for diagnostic performance. And so in particular for the 1 to 1.5-fold ratio here, we've annotated the chart to show an infinity symbol where they were not able to measure, an LODR was not able to be estimated for that ratio for that particular laboratory. And also I'll note that our LODR results, which I'm showing here, were also in agreement with the AUC statistics that we calculated across these laboratories. We also looked at the ratio bias across the different laboratories. We define ratio bias as this term R sub m, and this can be attributed to the mRNA fraction difference across samples, which I described earlier. And so really, if the nominal subpool ratio that you have, right, say this is, a 4 to 1 ratio, the empirical ratio that you should measure across the sample should still be 4 to 1. But again, if there's that mRNA fraction difference, you will have this RM factor that would adjust that ratio. And so if your samples did have the same ratio as you intended, then RM would be equal to 1. And so what we're looking at here in this figure is comparing the RM values across the different measurement processes, and also comparing them to a value that was measured empirically in this paper by Shippey and colleagues in 2006, where they actually measured the mRNA difference between these commercially available human reference RNA samples. And one can see that the ratio bias is actually highly variable amongst experiments, but also there's very large standard errors for each of these measurements, which gives us an indication that the mRNA fraction actually isn't the only factor that's contributing to these ERCC ratio biases. The mRNA enrichment protocol, which is a protocol-dependent bias, not just this inherent mRNA fraction bias that would be a property of the samples. So this protocol-dependent bias is an issue here. And so we explored this further by looking at the ERCC controls, and they revealed that there is a protocol-dependent bias in these experiments from poly-A selection, and it actually affects the ERCC controls because of they have very short poly-A-tails. And so what you're looking at here in this heat map is each row represents a different ERCC control that was measured across all 12 laboratories. And then the colors here represent the ERCC effect, which is essentially a residual from the signal abundance plot. So we intended to spike in a particular amount of ERCC molecule, and we got a signal that was higher than we expected or lower than we expected. And so if it's in the red range of color, it's higher than expected. If it's in blue, it's lower than expected. I will note that all ERCCs that were spiked in actually came through at lower than expected signal, given the abundance that was spiked in. And again, that's because of the short tails across all of the ERCCs. But then there are also particular ERCCs, specific ones that had more of an issue than others. And so another way to look at this, and one can see that it is clearly an issue for the poly-A enrichment kit that was used at labs 1 through 6, and less of an issue at labs 7 through 12. Another way of looking at this data is to look at each ERCC control faceted in this grid here. And we can again see that the protocol biases vary across individual ERCCs, but are consistent for a given protocol. And I'll zoom in on a couple of these examples. So for example, here, looking at these particular controls, we have ERCC number 51, which was didn't have dramatic differences between the different mRNA enrichment protocols. But then ERCC 46 and 44 clearly had specific effects were produced because of the poly-A selection process that was used in labs 1 through 6, which are shown here from left to right, the circles. And then again, here's another example where we can see that ERCC 116 was heavily affected by the poly-A selection process, and ERCC 113 was able to make it through actually a little bit better than, make it through the process. The recovery efficiencies were a little bit better than expected. So given this information, it's really difficult to be able to say that one could use the ERCCs for any type of normalization in these experiments. One would have to do a validated experiment to show whether or not ERCCs could be used to normalize data. But because we have this protocol-dependent bias, it precludes that type of application. And so to summarize results of the ERCC dashboard publication, we were able to develop ratio performance measures for any technology platform and any experiment, providing information on diagnostic power, our novel LODR metric, and technology variability and bias. We were able to do a comparison across experiments and quantify the mRNA fraction differences between samples, which is a powerful use of the ERCC controls. And we also observed these protocol-dependent biases from the ERCC controls because of the fact that they have very short poly-Atails. So now I'll close this lecture on the ERCC 2.0 effort, where we're building an updated suite of RNA controls. We've been recently approached by industry and academia to build new RNA controls as technology progresses. And so we hosted an open public ERCC 2.0 workshop. Our report from the workshop and presentations are available online. And all interested parties are welcome to continue to participate in the ERCC 2.0 effort. We're soliciting sequence contributions and also participants in our multi-laboratory analysis. Target controls that we're planning to develop in the ERCC 2.0 effort include new and improved mRNA mimics to improve upon the original ERCC controls. For example, possibly increasing the length of the poly-Atails. And people are very interested in having transcript isoform controls, and so we're working to develop those. And there's also interest, especially for our clinical applications, in microRNA controls. So during our process of developing the ERCC 2.0 controls, our intention is to validate these controls by testing these 3 RNA control hypotheses. First is that the RNA controls behave as mimics of endogenous RNA in assays. Second, the RNA controls do not interfere with the assays of endogenous RNA. And the third hypothesis is that these first 2 hypotheses will be valid in any commonly used RNA assay. And this is where the multi-laboratory testing using multiple platforms will allow us to validate this hypothesis. And so in summary, I've gone over today a little bit of background on gene expression experiments. I've given you an overview of work we've done with the ERCC RNA spiking control development. I've presented to you the new ERCC dashboard analysis tool, and I hope that folks that are interested will join us in our ERCC 2.0 effort to build new RNA controls. And with that, I'll close and acknowledge all of my collaborators who I couldn't have done any of this work without. And if you have any questions or would like more information, please feel free to contact me. Thank you.